Hello friends, in this video I'm going to take this very important topic, reciprocal regulation. What do you mean by that? First tell me, can two opposite pathways occur together in a cell? In a same cell, can two opposite pathways occur together? No, they can never occur together. Like example, glycolysis and gluconeogenesis. Glycolysis is breakdown of glucose. Gluconeogenesis is making glucose. They are two opposite pathways. They will never occur together. Body has mechanisms to prevent this. What will happen? Let's say, see my hands. Let's say these are two opposite pathways. If this is activated, this will be inhibited. And if this is activated, this will be inhibited. Like this will occur in the body. And this is known as reciprocal regulation. When one is activated, other is inhibited, and vice versa occurs. So let's see how this reciprocal regulation occurs in case of glycolysis and gluconeogenesis. Now, I am first going to take fed state. Tell me, in fed state, what you want? Glycolysis or gluconeogenesis? In fed state, obviously we want glycolysis, right? So, in fed state, one enzyme, PFK2 enzyme gets activated. First, tell me, what is PFK1? PFK1 is the rate-limiting enzyme of glycolysis. PFK1 is forming product fructose 1,6-bisphosphate. So it is very easy. PFK1 is forming fructose 1,6-bisphosphate. PFK2 will be forming fructose 2,6-bisphosphate. So this PFK2 will form fructose 2,6-bisphosphate. And this PFK2 is only active in fed state, right? Now this molecule, fructose 2,6-bisphosphate, is doing two things. The same molecule, this is the reciprocal regulator here now. It will activate glycolysis. But on the other hand, it will also inhibit gluconeogenesis. See the enzymes here. Fructose 2,6-bisphosphate will activate the rate-limiting enzyme PFK1 of glycolysis. And on the other hand, you know the counterpart enzyme of PFK1 in gluconeogenesis is fructose 1,6-bisphosphatase that is inhibited, right? So in fed state, this is happening. Now in fasting state, PFK2 is not active. So fructose 2,6-bisphosphate is not formed. So opposite will happen. Glycolysis will be decreased and gluconeogenesis will be activated, right? Now I'll ask a question. Let's say there is no acetyl-CoA. But there are lots of pyruvate molecules present. At this time, gluconeogenesis will occur or not? And this was my first question. My second question is, there is only one molecule of fructose 2,6-bisphosphate and there are 1,000 molecules of glycerol, and in this condition gluconeogenesis will occur or not occur. For solving such questions, I will tell you a formula. The formula is that regulator is always more important than substrate. Regulator means activator or inhibitor. Activator or inhibitor is more important than substrate. Let's see the question number 1. In question number 1, acetyl-CoA. What is acetyl-CoA for gluconeogenesis? I told you it is activator of very first step of gluconeogenesis. So acetyl-CoA is the activator. Now pyruvate is the substrate of gluconeogenesis. So you know more important is activator. So when activator is not there, then gluconeogenesis will not occur. Because activator is not there, enzyme is not active. Doesn't matter how many substrates are present, enzyme is not active, reaction cannot occur, right? And second question, there is one molecule of fructose 2,6-bisphosphate. I just told you it is a reciprocal regulator and it is inhibitor of gluconeogenesis pathway. And on the other hand, I told you glycerol is the substrate of gluconeogenesis. So again, now regulator inhibitor is more important than substrate. When one inhibitor is there and the enzyme will be inhibited, pathway will not occur. In this case also, gluconeogenesis will not occur because that one molecule of inhibitor will inhibit the enzyme. Again, enzyme is inhibited. Doesn't matter how many substrates are present, reaction cannot occur, right? Now see here I have written initial reactions of glycolysis. Glucose to glucose 6-phosphate, then fructose 6-phosphate, then the rate limiting enzyme of glycolysis PFK1 and fructose 1,6-bisphosphate is formed. Now, what happens in fed state? In fed state, there is lots of glucose. So there's lots of glucose 6-phosphate. So there's lots of fructose 6-phosphate. When lots of fructose 6-phosphate is present, mainly it is taken up by PFK1 enzyme forming fructose 1,6-bisphosphate. But because we have lots of fructose 6-phosphate, so PFK2 is also activated. When PFK2 is activated, it will form fructose 2,6-bisphosphate. I told you fructose 2,6-bisphosphate is a reciprocal regulator. This will activate PFK1. So can I say indirectly PFK2 is activating PFK1, right? And this fructose 2,6 on the other hand will inhibit gluconeogenesis pathway, right? And now there is one enzyme in the opposite direction. Fructose 2,6-bisphosphate gets converted to fructose 6-phosphate, and here we are going to remove phosphate from number 2 position. So name of enzyme is fructose 2,6-bisphosphatase. Take the name of substrate and add phosphatase in the end. So that is the name of enzyme fructose 2,6-bisphosphatase. This enzyme fructose 2,6-bisphosphatase is normally not active. It is active in some cancerous mutation, right? Now, before moving further, let's revise because otherwise you will get confused, because we have done one molecule fructose 1,6-bisphosphate. We have done one enzyme fructose 1,6-bisphosphatase. Then the molecule fructose 2,6-bisphosphate. Then the enzyme also we have done fructose 2,6-bisphosphatase. See, when I write phosphate, so it is a molecule. If we write phosphatase, that is a enzyme. So let's see where have we done all these 4 things. Fructose 1,6-bisphosphate, the molecule, this we have done in glycolysis. Fructose 1,6-bisphosphatase enzyme we have done in gluconeogenesis. Then fructose 2,6-bisphosphate, the reciprocal regulator. This we have done in reciprocal regulation. It activates glycolysis, inhibits gluconeogenesis, formed in fed state. And this last one now, fructose 2,6-bisphosphatase, I told you it is not there in some normal situation. It is present in some cancerous mutation, right? It is present in some cancerous mutation. Let's see what is that cancerous mutation. But before that, tell me, if this enzyme is active, then what will happen to glycolysis? So this is my question. If fructose 2,6-bisphosphatase is active, then what will happen to glycolysis? See this enzyme, fructose 2,6-bisphosphatase. When this enzyme is working, means the substrate will be converted to product. What is the substrate of this enzyme? The name of enzyme tells you the name of substrate. That is fructose 2,6-bisphosphate is a substrate, and fructose 6-phosphate is the product of this reaction. So what will happen? More and more of substrate gets converted to product when this enzyme is active. So the substrate will decrease, fructose 2,6-bisphosphate decreases. But on the other hand, fructose 6-phosphate, the product of this reaction, that will increase, right? Now we have to see the effect on glycolysis. That means we have to see the effect of rate limiting enzyme of glycolysis. If rate limiting enzyme of glycolysis decreases, then glycolysis decreases. If it increases, then glycolysis increases, right? Now, what is this? Fructose 2,6-bisphosphate. What is this for glycolysis? This is the activator of glycolysis, which is decreased. Activator is decreased, right? And on the other hand, fructose 6-phosphate is the product of glycolysis, which is increased. I told you the formula. Which thing is more important? More important is activator is more important. So when activator is decreased, then this enzyme will be decreased. So answer to this question is glycolysis decreases. Now let's see what is that cancerous mutation in which this fructose 2,6-bisphosphatase is activated and glycolysis will be decreased. Let's say there is some cell in our body, in a normal person body, there's a cell. This cell just replicated, DNA replication occurs and the new cell is formed. By chance, during this DNA replication, some wrong mistake occurred and the newly formed cell is having some cancerous mutation, right? Let's say it is having. Now, this thing is not allowed to do cancer in the body because we have tumor suppressor genes. So, when such a mutation occurs, immediately tumor suppressor genes will be activated. And you know, one of the main tumor suppressor genes is P53, also known as guardian of the genome. So, this P53 gene will be activated. By transcription and translation, it will be forming its protein TP53, tumor protein 53, right? This TP53 will induce another protein TIGAR, T-I-G-A-R, TIGAR. I will tell you the full form later. Right now, just look at this much, that TP53 protein will induce another protein, TIGAR, right? TIGAR has enzymatic activity of this enzyme fructose 2,6-bisphosphatase. So what will happen to glycolysis in this newly formed cancerous cell? What will happen to glycolysis? Glycolysis will be decreased. Now when glycolysis will be decreased, then there is no ATP in the cells, because when glycolysis decreases, then obviously the other things, link reaction TC, ATC will also not occur, energy will not be coming. So when there is so less energy in the cell, then one very good thing has occurred, that replication of this cell cannot occur. Obviously, we don't want this cell to replicate, otherwise cancer will occur in body, right? So replication is not occurring in this cell. Now, when replication not occurring, ATP is not there, then this cell is sitting silently, sitting in a dormant state. Now, in this dormantly sitting cell, all the repair mechanisms can come and they can try to do the repair. So, replication not occurring, but repair is allowed. All the energy which is there in the cell is diverted for repair. Little bit energy there, that is diverted for repair. Now, in this is like a time given to the cell that repair can occur in this time. If repair occurs in this time, then it is very good. TIGAR has done its work. If repair occurs, then TIGAR has done its work. It will be going back. P53 will be again inactive gene right now, and it is a normal cell. But if in this time given repair does not occur, then this TIGAR will induce apoptosis of that cell. It will kill that cell so that cancer does not occur in body. So now see the full form of TIGAR. TIGAR is TP53 induced because it is induced by TP53, and this protein is regulating two things in this cancerous cell, that is glycolysis and apoptosis. So the full form is TP53 induced glycolysis and apoptosis regulator, right?